Hello, so my name is Israel. I am 8-Bit Lookbook on Instagram, and we are going to study for lipid phosphorylation for biochemistry exam that is up and coming. So the first thing I use is GoodNotes, and what I am going to do is create a set of flashcards. First I type them out, then I go back and draw on them so I could memorize some of the processes. So first, let's create these notes. New notebook. It's chapter 19. Create. Now, what I like to do is I use a previous sample. I don't know why this one's blank. It should not be. But I like to use a previous example and I mimic the format. So Control Command Copy, Command V. The problem with GoodNotes is that it does this. So now I have to figure out why. Control Z, Control Z. Copy. Paste. There we go. So this is chapter 19. And this has to be undone. As much as I like GoodNotes, it does have its problems, which eats time into my studying. So for example, this, I have to I have to manually do everything, which is not effective for studying, but GoodNotes, if you're watching, can you please fix this? Because it is quite cumbersome. And that was my Pomodoro timer, which I'm going to have to reset. And then Control Copy and Control Paste. So now that this is created, I'm going to close this out. Go back here. Go back here. Chapter notes. Whoops. PowerPoint and lecture. I'm going to open the lecture. And scroll back up to the top. Then close this one out. Close this out too. Chapter 19. So this is where my madness begins. I am going to retitle this, re-edit this, oxidative phosphorylation. Doesn't help that I don't know how to type. Oof, this is a big word. Okay, and then I review some of the learning objectives so I can stay on track on what I need to know from these PowerPoint slides. Go back, scratch these out as I go along. So understand that the intermitochondrial membrane is impermeable to protons and other ionic substance. The inner mitochondrial membrane is permeable say impermeable to protons and other ionic substances. Understand the complexes 1, 2, 3, and 4 transfer electrons between redox centers that can undergo reversible 1 or 2 electrons reduction. Understand that the electrons donated by NADH and FA, I wonder if anybody ever calls it by its actual full name, are ultimately transferred to O2 understand the chemiosmotic theory. Understand the structure and function of ATP synthase. Let's correct that. Know the PO ratio, P/O ratio. Understand the NADH shuttles. Understand how oxidative phosphorylation, this word, phosphorylation, you would think I would know how to type it by now, is regulated. Okay, so I repeat, the goal of doing these objectives on the cover is so that I can go back and highlight the ones that I've reviewed and am fairly confident with, and leave the ones that I don't blank. So now it's time to create some of these flashcards. So oxidative phosphorylation, ATP synthesis, is driven by the transfer of electrons to oxygen Electrons donated by NADH, FADH, reduction of O2, H2O, mitochondria. See, the problem with these slides is that they're not very informative, so I have to go back and figure out how to phrase a question that will make me think on how to answer this. So, oxidative phosphorylation, that is why I use the book in tandem with the notes to try and figure out what exactly she was trying to say. Because clearly, I am not a mind reader. And her notes, her lectures, are particularly very uninformative. So the first, ATP synthesis driven by electron transfer to oxygen. So let's figure out what she was trying to say here. I mean, very clearly what she was trying to say, but oxidative phosphorylation. So I can't really show the book because copyright, you know? But I can sure as heck try to figure out where this is in relation to that. And what usually helps me is that I look at her PowerPoint. Oh, now she wants to put a bunch of words. Okay, so that backfired. So let's retrace. Okay. Oxidative phosphorylation. Well, let's come back to this. If I don't understand it now, maybe I'll understand it later. So I make a note to myself on the flashcard. Review slide 3. Something else that GoodNotes should probably do is work on its, I know it's not the goal of GoodNotes, but they should keep it updated, is work on how quickly you can format this. Review slide 3, because right now it's not really telling me much. 4. Actually, 4 is pretty explanatory. So let's go to slide 4 and let's create that. So let's just slide down. Let's go over here. No, I do not want that to be my default template. So let's see what I mean. Let's take away that and let's save this. Save as default. Let's delete this. Okay, let's go over here. Now let's go back. There we go. So first slide. Really seriously, this is, let's click here, click here. What is photophosphorylation? What is can't even say that. What is photophosphorylation? True. It is ATP synthesis that is driven by light, performed by chloroplasts. It's the reduction. It's the oxidation, my bad. Oxidation of H2O to O2 and ADP plus is the electron acceptor. Requires light. Light is required. So once I create one of these flashcards, I go back and I draw on them using my iPad. Therefore, I remember what exactly it's trying to say a little bit better than as if it's just pure text, because pure text doesn't really help me much. Okay. Fundamental similarities, electrons flow. This, I'm going to do a Venn diagram, and that's a whole lot easier if I just draw it out by hand. What are similarities? Between photophosphorylation and I assume it's oxidative phosphorylation. Photophosphorylation and oxidative phosphorylation. Let's stretch this out. Let's put this here. These words are too big, so they were probably not going to. Oh, it did fit. Electrons flow through a chain of redox intermediates. So, electron flow of redox intermediates. Exergonic flow of electrons coupled to endergonic transport of protons. Transmembrane. Transmembrane flow of protons down their concentration gradient. specific protein channels. Protein channels provides free energy for ATP synthesis. Provides free energy for ATP synthesis. Okay, so let's go back and review this. Understand how the intermediate, we haven't answered that, we haven't answered that, we have not answered anything, so these three slides have been, safe to say, a waste of time. But I write them down anyway, just like go back, review, because I believe they're more like highlighted levels. So let's go back. So chemiostatic theory, I know that's one of the things that I have to do. Understand the chemiostatic theory. So there, this is the first thing that we're gonna do. And we're just gonna create a line. So when I go back, I know that I have to do that. That we've created a flashcard on this, and I'm deep diving. So whoops, let's Command Z. Let's bring out our type box. What is the chemiosmotic theory? What is the chemiosmotic theory? Center that. Another thing that I would like for group notes is that it would kind of clip into place if you want to center something, as opposed to like me having to figure out where the center is. It would help with my OCD madness that I like to have things centered in the middle of the card. So let's go back. Chemiosmotic theory. Transmembrane differences in proton concentration are central to energy transduction. So what does that mean? Let's go back to the book. And let's keep scrolling until we find chemiosmotic theory. Smotic, whatever. Electron transport chain. How in the world? So I keep most of my books digital because, one, I like reading like this. I've gotten used to reading while looking at a screen as opposed to reading down where my head, I have a big head, so I could feel the weight on my neck, and this kind of reduces it. Not that I have the best posture, but kind of does. So let's keep scrolling. Okay. Searching for the chemiosmotic theory. And if the book doesn't cover it, a simple Google search always helps to know what exactly it's saying. Because that slide doesn't really explain much. So let's do this actually, because this is taking a little bit longer than I thought. ATP synthesis, chapter review. Are you kidding me right now? There we go. Okay, according to the book, the chemiosmotic model is the electrochemical energy inherent in the difference between proton concentration and the separation charge across the inner mitochondrial membrane. So it drives the proton. So it's a proton motive force which drives ATP flow passively back into the matrix, a proton in ATP synthase. So what is a chemiosmotic theory? Let's break this down into even more simpler terms. Let's Google search this. Chemiosmotic, whatever. Okay, so according to Google, the chemiosmotic theory suggests that essentially the most ATP, I'm sorry, that was wrong. The chemiosmotic theory suggests that most ATP in respiring cells comes from an electrochemical gradient across the inner membrane of the mitochondria by using energy of NADH and FADH form of the breaking down of rich molecules such as glucose. Okay. Okay, so now looking at this picture, it makes a little bit more sense that I'm seeing that there's a negative charge inside of the membrane, and the outside there is a bunch of positive protons just hanging around. So, oh, and here are the four complexes. 1, 2, 3, 4, yeah. Okay, so it's slowly starting to come back to me. So the difference between these two causes ATP to be generated in layman's terms. So let's reword this so that I can remember. ATP synthesis is due to, no, not due. ATP synthesis can be contributed to the difference in charge between the inner mitochondria and the outside cytoplasm. Proton concentration. So ATP synthesis can be a contributor to the difference in the charge between the inner and outer. Okay, let's reword that one more time. ATP synthesis can be contributed to the difference in charge between the inner and outer membrane. Outer membrane, also known as concentration gradient, aka concentration gradient. My answer's right here. Okay, another little nitpick about GoodNotes is that they should make this question line right here visible so I don't have to squint. I do like keeping everything in dark mode because it's softer on the eyes, but this line has to be a little bit more easier for the eyes to see. So for this particular slide, when I do this, I'm going to go back, draw on this, that there's a concentration gradient that is from protons hanging around the outside of the membrane and then a negative charge inside of the membrane, which is causing the inward flow. Energy of E flow is stored as electron flows potential. This is a IPT synthase complex. Okay. Kind of sort of got it. But again, this is just the step 1 of reviewing, and then step 2 will be drawing it out and really hammering it home so I can do well on this exam. So let's go back up here. Understand the structure and function of ATP synthase. Okay, so here, this membrane. So what's next is this is the my back. So what's next is so here in slide 7, we have a diagram of the mitochondria itself. Mitochondria is the powerhouse of the cell? Yeah. Okay, so we have the mitochondria, we have the outer membrane, the inner membrane, which is impermeable to most small molecules and ions, including the protons. It contains the respiratory electron carriers, which is the complex 1 through 4, and ADP to ATP translocase, ATP synthase, and other membrane transporters. The matrix contains pyruvate dihydrogenase complex, the citric acid cycle enzymes, fatty acids, beta oxidation enzymes, amino acids, DNA ribosomes, many other enzymes. So what do I get from here? What do I get from this particular slide? There are 3 main components. What are the 3 main? What are the 3 main components of the mitochondria? I'm not sure if this is a component, but the way I interpret this diagram is that's what it's saying. So let's go back. My dog is right next to me, and I could hear her stomach just growling. Even though she has food downstairs, she wants to be in here, which is fine. But I will say that the most annoying sound in the world is when she is grooming herself while I'm studying. That sound just drives me nuts. Or when she has finished eating and she's cleaning her mouth in my room and makes that like masticating sound, that also is a little bit, just a tiny bit irritating. Okay. Oh, what are the 3? Okay, so here, diagram is going to go, on this slide, there's going to go a diagram of the mitochondria. We are going to create the outer membrane. Outer membrane. And then let's create a new one, the inner membrane. And then the matrix. because I am half insane and I like having some sort of like structure. So the outer membrane. Let's create a new text box here. And let's drop down. I wish, here's another note for good notes. I wish that I could just type in the size that I want as opposed to clicking through this. Okay. The outer membrane, permeable to small molecules and ions. So permeable to small molecules and ions. Don't know why I wrote it that way. Okay, that's all the space that I need. Let's go back this up. Inner membrane, impermeable to most. So impermeable. Let's drop this down to 25. Impermeable to most small molecules and ions. Includes H+. This is important in where the complexes 1 through 4 reside, and these are the ones that pump out the protons. This is also important. ATP synthase is here. And that's about it. That's what I care about right now. The other things are important, but I don't feel like they're going to be on the exam. And let's drop this down. And in the matrix, there is the pyruvate dehydrogenase complex, also known as PDH. There is the citric acid cycle enzyme, citric acid cycle enzyme. Fatty acid beta oxidation enzymes. Fatty acid oxidation. Wow, that was terrible. Oxidation. Oh wait, beta. Beta symbol. Whoops. Beta oxidation. Enzymes. You can see why it's the mitochondria is the powerhouse of the cells. It's carrying out all these energy-inducing enzymes. amino acid oxidation enzymes. It's just generating so much energy. So yeah. Okay, so I think I am done with this slide. Let's create a new one. Here I'm just gonna delete this. Let's create a little bit more space so when I do go back and sketch on this. Let's create the space. So here, let's go back here and let's reduce here to here. So this is spaces for drawing and highlighting. Okay, Na can only transfer a hydride. Wow, my handwriting is terrible. So if I wrote it down in lecture, it means that it's probably important. So what can NAD pH transfer? Only hydrides. Don't know if I'm going to make a drawing about this one, but don't think so. What can flavins transfer? Flavins transfer. One or two electrons. One or two electrons. Okay, so mechanisms to transfer as a hydrogen atom or transfer as a hydride ion. Maybe we'll get it in the next slides. Ubiquinone. What is this trying to say? Oh, the reduction. It's reducing. So got it. Okay. So here's a ubiquinone. It has no added electrons, but then it comes this hydride with its electron. It binds over here. And then it repeats the cycle, so, okay. Gotcha, so 1, 2, 3. 1, 2. Don't think it's important. Son of a mother, what is this trying to say? Figure 19.4a. Okay, so this is where I'm going to call it an endpoint for this study session because I do have to go to class and then I'm going to pick this up later. So with that, I will bid you adieu.